Hello students, welcome to biology class. Today I'm going to discuss about practical examination. As we all know, we have practical examination for 30 marks, but as per the revised syllabus, there are two terms, isn't it? So for term 1, you will have practical examination for 15 marks, term 2 also 15 marks. Okay, so this is a new scheme for your practical examination. Three sections will be there for your practicals: part A, part B, part C. Okay. In part A, one major and one minor experiments are there for 7 marks. Part B, there are spotters. Part C is viva voce. Okay. So in part A, major experiment is isolation of DNA, and minor experiment is slide preparation. You have to prepare a temporary mount to observe pollen germination. Then in part B, there are 5 spotters. First one, flowers adapted to pollination by different agencies. Second one, identification of stages of gamete development, that is testis of TS of testis and TS of ovary. Then third one, meiosis in onion bud cell or grasshopper testis. Then fourth one is TS of blastula. And the last one is preparation of pedigree analysis for any of the traits. Okay, the 5 traits are given. Genetic traits are given: rolling of tongue, blood groups, earlobes, widow's peak, and color blindness. Okay, and part C, that is section C, is viva voce. You will have viva voce from practical record and investigatory project. Okay, so this is your new scheme for practical examination. So you have to start writing the record book from now itself. Okay, so I will explain the order. Okay, in the middle of the first page you have to write term 1, and the first experiment is study of adaptation of flowers for pollination. You avoid introduction, no need to write the introduction. You can start writing from aim, then requirements, procedures, then maize flowers and all the points, then salvia flower, then bignonia flower. Okay, and you can write, you can draw these diagrams in the left side of your record book. That means on that blank part, these two diagrams and these two diagrams. Okay, and you can avoid the viva voce question, no need to write. That's the first one. And second one is study of gamete development and mammal, no need to write mouse. Avoid introduction, start writing from aim to study and identify the stages of gamete development in mammal. TS of testis and TS of LS of ovary, write requirements, then procedure, observation, TS of testis, VS of ovary. Draw these diagrams on the left side, write precautions, then avoid viva voce questions. This is the second experiment. Third one, study of meiosis in the floral buds of onion. Okay, no need to write and testis of grasshopper. We are writing only the first one, study of meiosis in floral bud of onion. Avoid this page, no need to write this page, this introductory page. And start writing from here, that means from aim to study meiosis in onion bud cell through permanent slides. Then write requirement, procedures, observation, meiosis 1 and meiosis 2 and precautions. Draw these diagrams in the left side. Okay, no need to write experiment 12 too. We are writing only the first experiment from this part. Clear? Then avoid these pages. Next experiment is study of blastula. Avoid introduction, start writing from aim, requirement, procedure, precautions, observations, precautions. Avoid viva voce questions. Draw these two diagrams on the left side. Draw these two diagrams on the left side. Then the last spotter is study of pedigree chart. Avoid this page, no need to write the introductory page. Start writing from experiment 15.1, that is aim, to study prepared pedigree charts of genetic traits such as rolling tongue, blood groups, widow peaks, and color blindness. And write the problem: inability to roll tongue, first one, and second problem, widow's peak, third problem, color blindness, and third problem, color blindness. The question is here and its answers are here. Avoid viva voce questions. Clear? You can draw the pedigree chart on the same page, that means on the ruled page itself. Clear? And this is the next experiment, study of pollen germination. Here also you can avoid this introductory part and you can start writing from aim, then requirements, procedure, observation, precautions, and this diagram on the left side, and avoid this 5 hours question. And the next experiment is isolation of DNA from plant material. No need to write this introductory part, and start writing from aim and write requirements, preparation of solution, procedure, observation and precaution, and this diagram on the left side. Okay, so that's about the record work. Okay children.